Okay, so moving on to the next session, so the panel discussion. So again, we've heard a lot over the last 2 days, both thinking about applications, but also around some of the challenges in standardization, and as the field moves forward and starts to populate VOC atlases, trying to get a bit of a sense of what would be valuable to have in the atlas. So really excited to have the panel discussion where we have James from University of Warwick, Brooke from PNNL, Iain from University of Manchester, and Ben from the University of Bristol. So if you're all able to unmute and show your cameras, we can kick off the session. And while you're doing that, again, we want this to be very interactive. So any of the attendees, please feel free to send questions into the Q&A, and we can make sure that the panel is able to field those questions. What would be helpful just to kick us off is maybe for each of you to give an introduction, who you are, and some of your experiences in VOC biomarkers and breath detection. So why don't we kick it off with you, please, James?
Do you know something? I knew you were going to pick on me, Billy, first.
You're top left on my camera. So
okay, so I think as Billy introduced you, my name is James Covington. I'm from the School of Engineering at the University of Warwick. So I've worked with Billy, or known Billy, for I think 20 years, might be 25.
20.
I think since the last millennium.
I hope it's 20.
So we worked together a long time ago when we started working on VOC. So in essence, I'm a gas sensor specialist. So I work in developing gas sensors, and then I've got into breath analysis and VOC. So I also work on international standards for odor devices. So I chair working groups with the IEEE to develop standards odor devices. There is, as a quick plug, there is plans to do a breath analysis standard through the IEEE, and if anybody's interested, they can contact me. So it's really interesting what Billy says. So I think 10 years ago, I had this, as a gas sensor person, I had this thought, oh, it's easy. I can go pick up, I don't know, I go out to the field and I can detect odors from, some rotting onions or something. How hard could breath analysis be? 10 years later, I've kind of learned my lessons that the variances in breath are significant. And I think the biggest thing that I didn't take into account is the variances of the people collecting the samples. And at the end, you pick up that the nurse or the practitioner is more important almost than the technology that you're using to detect it, because they are usually the wide variance in it. I also learned a lot about where you collect your control samples versus where you collect your real samples. And I'm sure that there are plenty of papers that show that they've got two separate rooms, and one room is near a polluted street and one isn't. So I think my kind of my care and attention on doing breath analysis has gone up a huge amount over the last 10 years. And now kind of my final point is it's amazing what people will smoke before they go do their breath analysis. And some people do smoke some quite exciting products and give very interesting spectra. Shall we leave it like that?
Excellent. Thank you, James. Iain, over to you. Please, could you introduce yourself and talk a little bit about your research background and interests? Over to you.
Okay, I'll start again. Hi, I'm Iain. I'm from the University of Nova Gorica in Slovenia. I started out life as an atmospheric chemist and eventually got into breath research when I was at the University of Leicester, and then eventually the University of Manchester. And I still work closely with the guys at the University of Manchester who have ongoing studies, biomarker discovery studies related to respiratory diseases, breathomics. And right now I'm particularly interested in using breath in biomonitoring following exposure to air pollution. And I'm particularly interested, as I'm sure we'll discuss, in moving towards targeted quantitative studies. So yeah, that's my brief intro. And sorry about the microphone issue.
No problem. Thank you so much for that. Super helpful. Ben, maybe you could go next. Just a bit of background on yourself and your research interest, please.
Hi, I'm Ben Lacy Costello. I'm an associate professor at UE Bristol. I've worked in the field of disease detection in humans for around 20 years or so with various colleagues and collaborators, particularly in gastroenterology. I did, with some other colleagues, start up the Journal of Breath Research, which is why it's an IOP journal, because they're based in Bristol. And I did also publish a review on volatiles linked to the healthy human body. So I've got some interest in VOCs relating to breath research, etc.
Wonderful, wonderful. And the great thing about that paper as well was that it was a broader range of volatiles in different sample types as well, which I think is very relevant for the discussion later. Brooke, please could you introduce yourself and give a bit of a background?
Yeah, hi everybody. I'm Brooke Kaiser. I'm a biomedical scientist at Pacific Northwest National Lab, which is in Washington State here in the US. And my background is microbiology, so I'm interested in infectious disease, but got into breath research really, and have been working on a project for the last few years that's really interested in trying to understand what healthy human breath contains. So we talk a lot about, using breath as a diagnostic, but really that's predicated on sort of knowing what's quote unquote normal. For most of us, which as probably everybody on the call might imagine is kind of a challenge to define what is normal and what is healthy in lots of different populations of people. So happy to join today, and thank you.
Wonderful, thank you so much. So we had a call last week to kind of go over some of the topics that we might want to cover in today's session. And one of the things that struck me, Ben, was a comment that you made that we talk about standardization all the time. We go to conferences and it tends to be, a topic of discussion all of the time. And I think what's quite exciting to me is seeing, we saw some of the talks from the FDA yesterday, NIST today, MPL poster this morning. James, you're talking about some of the efforts around standardization there as well. So what I'd like to try and kick it off is to say, it's been talked about a lot. Why has there not been more progress made? And actually, do we feel we're now at a point where we will start to make progress with these different initiatives and institutions like FDA getting involved? So who wants to be the first willing victim of that question?
I'm going to start. It's because I'm doing this now for a different standard. I can almost tell you, I would assume that the problems within the standardization of the breath will be exactly the same as the standardization in ODA devices. And both the European groups and the US groups have tried quite hard to do this. But it's really been, the European group kind of collapsed at some stage of it because they just couldn't agree how to do it. So the problem you have is a lot of researchers who have done a lot of work within their field, and bringing them together to try and create a standard is phenomenally difficult. And the sort of thing about where, how you do it, whether or not you use gas-based standards in labs, whether or not you should be doing your, for a breath analysis, it might be, the standard test is you go to a hospital and use patients, and that is how you calibrate your machines. And there's a lot of tension between different people and opinions on how to try and achieve this. And it's one of the reasons why the kind of European work in this field kind of paused, because they just couldn't agree as a group about how to make this. It's not about them not wanting a standard. It's just that they can't agree on the standard.
Interesting. And Iain, maybe picking up on that theme, would love your thoughts and feedback on that to see whether or not there's enough of a will and impetus and momentum within the breath field to try and progress it, or whether or not similar challenges may crop up.
Yeah, absolutely. And it's definitely a will. We all want to create a standard. I would add to what James said. I completely agree. And I actually think that part of the problem is there's a lack of published data to support a lot of the different opinions that people have. And I'm saying it because I'm a proponent of publishing quantitative data. And of course, a lot of the biomarker discovery studies that have been reported in the past didn't, following an untargeted analysis, report, quantitative concentrations. Now, research proposals have moved forward and, trying to take things to the next stage, implying that the biomarkers have been discovered. But there are more biomarker discovery projects going on right now. And so, on some level, I think we as a community need to report mixing ratios, quantitative concentrations more, along with other often unpublished results like, related to background subtraction, that sort of thing. I'm saying this because earlier in the year, we published a review in Trends for Analytical Chemistry. And as part of that, we were trying to compare different results. And to start off with, looked at the most studied non-communicable diseases in asthma and lung cancer. And I was startled by how few papers I could find that had reported mixing ratios. And two problems cropped up when looking at the results, often reported in reviews following breathomics studies, is mixed direction of effect. Control groups sometimes have lower levels, sometimes have higher levels, but also that there was very little quantitative data available to properly scrutinize different studies and compare. I found for lung cancer, for example, only 2 or 3 examples of compounds which you could compare in terms of mixing ratios. And I think this is super important to compile, not only so that we start to build up a database that reflects normal levels in breath, that shows you the levels from different studies of different diseases, but also so we can compare different analytical platforms. Because of the compounds I looked at, one was 2-butanol, as I recall, 2-butanol. I was delighted that the authors had published concentrations, but it was clear that there were 2 different analytical platforms that had data related to them: SPEME mass spec, and it was silicon microreactor FTICR mass spec. And broadly, levels compared quite well, but I thought it was interesting that one set of results was a couple orders of magnitude lower than the other. And that could be analytical platform, it could be the sampling, it could simply be data pre-processing. But until we start compiling all of the quantitative information, we can't properly scrutinize the data.
Sorry, I've got a question for you, Iain. Sorry, Billy, I'm jumping in front of you.
Go for it.
If you were writing a standard, what is the bit of the standard? So if you write a VOC standard, you say we're going to test with, we're going to take a platform, we're going to test with these chemicals, we're expecting it to be able to achieve this and pass with this, and then you get the standard. So what would you do on breath?
What would you do on breath? Well, I think, and this is a problem that everyone cites, is of course, which compounds are you looking at? And it may be that we need to select, how was it phrased earlier?
The surrogates from
surrogates. Thank you. I couldn't think of the word surrogates. Yeah, I completely agree with that approach. But then a set of criteria on how you validate your test would be the first place to start, I would say. I mean, I don't think validating a method is simply showing that you can build a calibration curve. Testing a method for matrix effects. Of course, background, defining what an on-breath compound is fantastic, and it's been pushed forward recently, of course, we heard today. But what do you report? Background subtracted levels, which background? Those sorts of questions should be very well defined in any standard moving forward for any compound.
Yeah, and it's interesting the point you're picking up on. When you know the compound and if you quantitate, then actually it's less about standardization of a particular sample or analytical platform. It's more about standardization of the data that gets collected that therefore allows cross comparability as well. I think we had exactly the same experience when we were developing sensor technologies. We thought, brilliant, we'll go to the literature, we'll get reported compounds for lung cancer, functionalize it, we'll have a test. But as I said, there's no agreement in the papers and there's no quantitative information as well, which is why you need to go back to address some of those issues. And Ben, I'd love to turn it over to you. So what Iain is saying there about the lack of quantitative data there, when you were doing your review, I was wondering if you could comment a little bit further on what you saw in that regard, and maybe some other observations or challenges that you saw in that paper.
Yeah, I mean, there were various challenges, obviously. I mean, the data is quite disparate in terms of a lot of the studies we looked at, because we were looking at healthy humans, obviously, or trying to get a baseline, I think, to really compare what is healthy with what is diseased, because most people are funded to do projects which involve a small cohort of diseased and then some healthy individuals, and those healthy individuals might be people from the university or the company or whatever. So they're not really matched in any way to the diseased individuals. And we were just picking data really from those control groups, which were not designed or really thought about usually in the studies to any great extent, and usually very small numbers. But for that reason, you end up with a huge number of compounds, obviously, which are somewhat unverified. I mean, some of them didn't even exist, but they were published. So there was a degree of correction. There was like attributing CAS numbers, for example, things that would make it more useful for people in terms of how do we actually, because people just tend to report the NIST library, and depending on how many times that compound's been run, it can come back as maybe 3 or 4 different things. So those can cause issues because people tend to do studies and then want to sort of benchmark it against something. And I think from what I've heard from people, they do use the review, but I don't think it's what they really want, because it doesn't really contain quantitative data. It contains a lot of compounds which probably are very, you could argue that a lot of them come from exogenous sources, potentially. That we can't really exclude those from breath because they can be changed in the breath. But I think, there isn't much quantitative data there either. And that is a big issue if you're developing sensors or any type of systems. I think the thing that's limited standardization, particularly from my perspective, is that from an academic perspective, people find that standardization word is restrictive on them. They see them doing discovery science, etc. But we see the results of that is a lot of disparate studies which disagree quite extensively. But I think people also see standardization as limiting, new technology and bringing that online as well, which is probably problematic because people like to have a new, more sensitive GCMS, whatever, a better method for, collection.
Yeah, and I suppose it does depend again on exactly what we mean by standardization, because if it's more about standardization and agreement of the quality of data to be collected, that then is completely permissive of whatever sampler you want or whatever analytical platform that you want as well. Because I agree with you, you do need to have a bit of a balance of not shutting down avenues to find new things, but then how do you actually progress things forwards in a way where, you know, they are ultimately going to make it in the test, as the kind of team at FDA were talking about yesterday. And maybe, Brooke, could I bring you in there as well? Obviously, you've been doing your work at PNNL with DITRA, using GCGC high-res instruments. I was wondering if you could elaborate a little bit further on the work that you've been doing and how you're trying to tackle some of these challenges around identity and quantitation in that healthy cohort.
Sure, yeah. So, so we've been, as Billy mentioned, we've been looking at trying to understand what normal breath looks like. And so our approach, we're one of kind of 3 projects that's taking different angles at this problem, different cohorts of individuals that we're sampling from. Ours in particular had about 250 individuals that were sampled 6 times over the course of about a month if they showed up for all of their sampling appointments, which most of them did, thankfully. And so we have some longitudinal component of that. But, you know, the reality is there's a lot of data, and when you're generating that much data up front, our approach has been to not give anything names. We just kind of are blind to whatever, we call them features. And then once we start to, you know, take it downstream further and, you know, statistically analyze and try to understand what are those things that are more conserved among us, then that's when we go in and start to identify what some of those things are, you know, using, you know, NIST scores and library searches and expert review of, do we believe, you know, what this compound is. And so that's kind of been our approach. We don't do any, you know, checking against standards at this point. Part of that is because some of the things that we're identifying don't have good matches, they don't have good names, or they don't have standards that are available. And I think that that's, you know, getting back to kind of the big question here about standardization, that is another challenge. If you're finding things that you can't get as a standard that to check it against, of course, that's difficult. And just the reality of, you know, funding, maybe if you don't know that you're going to have to prove those things are the things that you say, you haven't budgeted in, you know, to do that side of the work. So I think moving forward, as we talk more and more about, you know, what does standardization mean and what will go into that, you know, thinking of that at the proposal stage of a project and understanding, you know, if I want to publish or if I want to be a part of, you know, adding my data into this database, what are the other steps that I'm going to need to do?
I think that's a really good point, because it's expensive to buy all the standards as well. So, you know, often there's a temptation just to move on to the next thing. But if we do want to kind of add a new brick of knowledge, and then I think being a bit more kind of prescriptive about what does it actually need to look like is very important. And I think one of the things that jumped out at me from Kavita's talk at NIST was, so some of the test gases that generate now, so there's kind of leveraging what has worked in the past. And, you know, Iain, you talked about background in atmospheric chemistry. Obviously, VOC analysis is very relevant. There are, James, talking about different standardization efforts, what's worked and what's not worked, and, you know, human factors, shall we call them. But obviously, you can look at adjacent fields, things like, you know, standard metabolomics. So do you think there's elements that we can pull from adjacent fields that we can apply to the breath field that would help in this exercise, be it again VOC analysis elsewhere or these adjacent disciplines? I'll open that up to whoever wants to go first on that one.
So I will let Iain have a thing in a second, but I would say that if we're only at the point where we're getting standards for VOC analyzers, and that in theory is an easier problem than doing a standard for breath, it could well be some time into the distance before we make it. And I actually was going to ask you a question of whether you think we'll make it before I retire.
Well, you're still a very young man, Jim.
That's very generous of you.
But, I would say yes, absolutely. But again, I think it comes back to one of the things I do find quite exciting about the field at the moment is where we do see, deeper involvement, from FDA, NIST, Gates Foundation, and this. Iain said as well, there's certainly a desire. So it does seem like there's an opportunity to crystallize the desire into tangible results here. So I'm certainly optimistic.
So I'm going to also ask the panel another question, whether or not they think that we should have standards for collection. Do you think we should have some example generic, simple approaches that new researchers could use if they come into the field? Because one of the things about a new researcher is they are often just hunting through the literature, trying to find a way of collecting breath samples. And whether or not we, as I was going to say, more experienced researchers, should find ways of supporting them by kind of creating some methods or something that they could start off with.
But I think this comes back to Ben's point. You don't want to be so prescriptive that you can't use new technologies, either on the sample collection side of things or on the analysis side of things. So my personal view is, if you are saying, here's what is expected from the data quality, be it ID, quantitation, etc., then people can use whatever approach that they want to use. So that's my kind of personal view. But again, I don't know if Iain or Ben or Brooke would.
Similar to different field where you have a minimum set of.
Oh, you've gone on mute.
Yeah, sorry, I'm having connection problems. I do apologize.
Right.
I was just saying, yeah, it's similar to different fields, right? Where you have a minimum set of criteria. And there is a method, an ESOL method or whatever, ISO method that is there and available to use. And you can use an alternative as long as you meet the minimum threshold criteria. Yeah. And that makes a lot of sense. And I think it's worth mentioning now, following what you said earlier, Billy, worth citing the Metabolomics Standards Initiative, which have already laid out some minimum criteria, well, certainly minimum reporting standards anyway, for metabolomics studies. Yeah, it's worth mentioning, if nothing else, because certainly some of that can translate into the Atlas database.
Yeah. Excellent. Anything to add, Ben or Brooke, on that point?
Yeah, I agree that, I think, like Brooke said, the cost is one thing that limits standardization. And I think possibly, the community follows money in terms of getting grants to work on specific diseases. And obviously there isn't a great sort of community in terms of let's try and establish what is a healthy breath profile. So we obviously require then organizations to do that, because there's some, there's a commercial reason for that. If you're at a late stage of developing a sensor for a specific disease, you need to have that level of standardization to make that product viable. But if you're just looking at finding biomarkers for a new disease to write a nice paper, which will, go towards your ref in your university, then maybe that standardization isn't close, or isn't as close in terms of necessity. But I think that maybe limits that people sort of working together from the academic community, the breath research community. But you can see that it's a much wider community than just people interested in breath research, for example.
Yeah.
I think again, learning. Sorry, Billy.
No, go for it. Go for it.
I just wanted to say, in response to something you said earlier as well, then, I think we can take something else from the metabolomics community, in that at some point there was a method that was laid out for reverse phase liquid chromatography, mass spec. And, we all know as a community that it's not necessarily the best method, but it was just decided upon that we'd use that method with that criteria, just so that we could come up with some sort of standard. And that's been quite successful. And people use a very similar method across research groups across the world. So, you know, perhaps we need to do that as a community and take a hit on trying to work out what exactly the best method is and just settle on a set of criteria.
That's interesting. So being prescriptive on, platform, be it sampler or analytical platform or method that runs on that. That's interesting.
Yeah, I think just something that I think is, I mean, I agree with it, and I think it's not to do with the inability to innovate. It's more that if you are, early researcher, you will make exactly the same mistakes as I made, Ben's made, probably Iain's made, and I think that's an inhibiting factor. If we can move people, educate them a bit further down the line, so that they're still already starting to get, reasonably quality data, some kind of level of standardization, etc., then I think they can innovate. But I think it's important to try and move people from these horrific mistakes that often happen when people start, and just, move them a bit forward.
Yeah, yeah. Excellent. Okay, so we'll kind of move on to the next part of the discussion, which is kind of thinking about, if we move towards standards, be it instrument or reporting standards. So it does sound like certainly just having data available to the community would be valuable, and we can think about that in the context again of, nominally healthy control populations, ensuring that the compounds are ID'd, they're quantitative, etc. So we'll move on to that section. We ran a poll through the day with a few questions to try and get a bit of a sense of how useful a database or an atlas would be to researchers, and a few interesting insights here. I'll kind of pick out a few key points to seed the discussion. So the first question in the poll was, what, if any, reference database tools used in research? And so NIST overwhelmingly is what is reported there. And there's a few researchers that have reported their own database, so maybe where they've run studies in the past and they have a bit of an understanding of what's present on breadth. And the next question was, would you use a reference VOC Atlas as part of your research? So 96% of people said yes to that. So I think that's really interesting. It shows, as some of the elements that we've discussed here, there is a need for a tool where you can see some of this data and get some of these insights. And the next question was looking at some of the things that would be useful to see in a database or an atlas. So a broad overview of literature and associated compounds and diseases. So 48% of the respondents said that would be useful. And the other probably more interesting one is, a large sample size of healthy breath data for comparison. 67% of people said that would be useful. So that in itself sounds like it'd be an incredibly valuable thing to put out into the community. And then two other questions were, how would you use VOC Atlas in your work? A reference, screening, identifying common VOCs and narrowing search for unique applications, confirm findings and shorten the phase when choosing next research direction. We would look at the most common VOCs by abundance and develop gas reference materials for components we don't currently have capability. I'd use it for biomarker discovery, patterns of disease, identifying interfering compounds for our studies, clues to important biomarkers for specific diseases. And then the last one, are there any features you might want to see in a database or an atlas? So pathologies and how they link to VOCs, endogenous and exogenous, so picking up on that point earlier. Curated with clear indication of source reliability, compounds, so CAS numbers, related diseases, correlation with other omic data, guidance on how to apply to electronic nodes, and then how some of those VOCs could be detected as well. So some really interesting points coming back from the poll respondents. So thanks everyone there for taking the time to respond. So we've picked up on some of these things already, but I was wondering if I could just ask, how valuable do you think it would be to have a database or an atlas that does link VOCs to different diseases and does have data that is downloadable, both for healthy, nominally healthy cohorts and disease cohorts. I'll pick Brooke at random on that one there. Is that something that you think would be of benefit?
Yeah, absolutely. I mean, I think that something that we have found to be a challenge is when we're comparing what we're finding to what people have published. You know, a lot of things are said, oh, it's, a diagnostic for this infection or this exposure. And it's identifying, a table of VOCs, and we look and we say, well, we see the same things in normal breath. And so how do you get from, where is that normal space versus, the abnormal? I think having this kind of a database to go to and have information there that you believe, that you're confident in, that has, information to back it up, that you can actually view yourself, would be a huge benefit, because oftentimes all we see in a publication is maybe a box plot comparing, control to treated, and it's like, well, I don't know. And so I think this would be, it would be a great tool to try to get further in differentiating sort of where does that normal range fall versus outside of normal.
Yeah, and if you have 1,000 features in your data set and only 50 samples, it's pretty likely you'll find box plots with some decent separation. Iain, do you want to pick up on that?
Yeah, absolutely. I mean, I think database sounds very useful. And the information that we're talking about is a bit like what's presented in the HMDB, right? The Human Metabolome Database, which has cards for specific compounds. But I think there are some things specific to breath, which mean that it would be very useful having a breath database. I mean, perhaps one of the problems we might face is we might have so many suggestions of what can go in. It's going to be difficult picking and choosing a short list. But for example, results from in vitro studies versus in vivo. There's a lot of data there that could go in. It might be a lot of work to compile it. But I think absolutely, as well as endogenous and exogenous. I've already, of course, voiced how quantitative data, compiling that somewhere would be fantastic. In terms of looking at the source of a VOC or the provenance or whatever, I think perhaps other information can go in as well. There's super interesting work about the kinetics between different compartments in the body, right? And so those coefficients could go in, Henry coefficients, that sort of thing as well, to try and rationalize your VOC might actually be a marker that can scrutinize between diseases. Yeah, so I think there's a lot of potential to pull the information in one place, and it would be fantastic.
Excellent. And I'll just pick up on the quantitation point again before I turn it over to James and Ben. So why do you think quantitative data has not been done before? Again, is it a cost complexity more than anything else?
I think it's largely, it's whether or not studies have been targeted or untargeted, and most biomarker studies are untargeted. And then to report semi-quantitative data after that, you have to make a few assumptions, which I think people aren't willing to go on a limb on and make. Fair enough. But perhaps we could think about the more like lipidomics approach. So metabolomics, untargeted, you might pool together a QC mix where you take a tiny amount of everyone's sample of blood, let's say, and then have a pooled sample. We can't do that with breath. The lipidomics approach is having one big standard with lots and lots of different lipids in. And there's potential there to quantify a lot of different compounds. So I think maybe we could learn something from the lipidomics community.
That's super interesting. I think some of the work presented by Mark's earlier and by this team again, I think is will be super helpful to us as we move towards more quantitation. James, from your perspective, do you think it'd be helpful to have that type of data? And the kind of maybe the additional twist I'd give you, you've talked about your sensor background as well, so you can think about this type of data being useful both for a biomarker scientist, but also us sensor guys, being told here's the thing we need to detect at these levels. So I was wondering if you could kind of comment a little bit on, would you see utility in having this type of resource?
So I have to say it was interesting. So for everybody else, we had access to a demo version of the VOC Atlas last week. And the first thing I did actually is to go look something up that I was interested in. So actually, there you go. As a thing, I actually already found it useful in terms of finding something out. I think, in terms of sensor technologies, there are molecular groups which are easier to detect, and as others which are a pain in the arse. As you know, Billy, as you've done sensor work yourself. And I think it was when I was kind of looking through the list, I was kind of flicking down going, oh no, 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 no. Oh yes, I've got a chance at that one. So as a sensor developer, I think that there are, if you're focusing on that aspect, you can go through your biomarker list, you can pick out molecules which you think you've got a chance to do. You can check the concentrations of those and see if you could, you have a chance of detecting them. I think, there's lots of discussion of background, but if you can't pick it up to begin with, the background is neither here nor there. If your sensor technology is not going to detect it at that concentration, then it's game over before you worry about the background environment. So I think that is a really useful tool. And in fact, I very much liked the up and down on the database and found that really useful. I strongly suspect you found loads of papers which say these low go up, and another set of papers that the same molecule go down.
It's exactly as Iain said. That's exactly what we find, and it's just like, well, how can these two things be true at the same time?
So I thought that was really interesting. So firstly, like I say, I actually used it to kind of give me some feel. And I also think if you are thinking about researching that field, it might tell you, oh, it's don't worry, don't bother. It might actually say your original study has been done to death. What you should be doing is thinking about the next step, or you should be thinking sidestep. And it's so difficult to do a kind of complete literature review on a certain disease and then look at those biomarkers that I think that would be really useful for people. If you can then boost that up with having the kind of raw data and kind of do some data mining, I think that'll be great. But I'm hoping it'll stop people doing repeated studies again and again and again, the small discovery style studies, and either move people to do larger studies where, they go, okay, these are our most likely candidates. So a little bit of pre-discovery for confirmation and then a big study. Or say, no, I'm going to leave that. I'm going to look at a different medical question because I can see that there's a ton of literature on the field on that. So from a census perspective, I think that's really where I'm seeing, gain for me as a researcher.
Excellent. And then Ben, from your perspective, having database or atlas of those types of compounds, love your feedback on utility of that.
Yeah, I think it's like, from my perspective, like trying to get systems that will diagnose disease. I've always found that we just don't have enough information about what is healthy, especially quantitative data. So we haven't ever had that in one location. We've had to search through papers, and as Iain said, it's frustrating because the data just isn't there for many compounds. We've tried to do studies to quantify compounds in stool for a commercial company, and trying to get isotopic standards for various things was prohibitively expensive even for them. So it's difficult to get this quantitative data, but that's why it's so valuable. I think from my perspective, when I was looking through the database, I was looking at how low the concentrations are of some of these things and thinking, well, actually, from a sensor perspective, a lot of these things are, we're not going to look to develop sensors for those. They really are very, very low concentrations. So that was one thing that struck me. Once you get past the first few compounds on breath, most of them are there at part per trillion, like even lower levels by the looks of the database, which is interesting.
Yeah, no, we observed the same thing. It was quite funny actually, because the visualization in the VOC Atlas at the moment, there's a kind of simple abundance bar graph. And I was kind of saying, well, it's a bit pointless because I say after the first few, everything is so small that you can't see the visualization. But I think that point on just being able to know the concentration levels is incredibly valuable if you're thinking about, again, targeting the specific VOC for an application. It does rule out quite a lot of sensor-based approaches. So we saw yesterday some of the work on indole and how it relates to changes in blood glucose that have been previously reported by Roche, for instance, but it is at very low levels, even though it is one of the higher abundance compounds there. So you're saying, well, actually, that's probably not going to be feasible to detect in a sensor-based solution. You need sample collect and lab-based analysis. So it is very fascinating to see. It does help rule out certain things as well as open up the possibility for what should be worked on. And the point about literature is quite interesting. I don't know if you all saw Julie's presentation earlier. So what we've tried to do in the VOC Atlas database so far, and again, it'll be available for everyone to use shortly. Part of it is looking at what has been reported. So, where we've scoured the literature, reported the compounds that were in the paper, directionality, is it up, down, unchanged? Sample type, is it breath or headspace? Again, there's questions of, as it's been said several times, more often not only don't you have the quantitative data, but you don't have a confirmed ID for that particular study in the VOC. So there's the kind of what has happened in the past. So my question is, how valuable do you think it is to grow that portion of the database that will help people just search for what's been done before, versus data that goes into prospectively, where again you do have agreed reporting standards on it's on breath, it's confirmed ID, it's quantitative, etc., and then users can download that, because both take resource and effort. And I was wondering if you had a bit of a sense of what would be more valuable and where we should put more emphasis as a community. Maybe Brooke, I'll start with you on that one, if that's okay.
Sure, yeah. Well, I've already complained about the literature, I guess. So no, I mean, that does offer, of course, perspective into what people have done, and there is definite value there. I mean, I think moving forward, as we have talked about standardization and we have talked about having more confidence in what some of these things are and understanding what is normal, I think that, having the data there to look at, probably if you have to choose one over the other, we all, we already are doing literature searches. And so maybe those are things we can more easily get to, but new data that you have vetted would be a great addition.
Mm. Anyone want to add to that? General agreement, disagreement, nuance?
The only thing I would like to ask, so I totally agree with Brooke, but I would like to add, if there is a disease that the only paper is a semi-quant or whatever, then it'd be nice to have that, because then it just helps you. So I think if you can if there's quality data there, have the quality data. But if there is absolutely no other study ever on that disease, then I would still like to have that one.
Yeah, fair point.
Yeah, I mean, I think that's right. And I think we discussed in sort of like the pre-discussion, like sometimes if you've got general consensus from the community about, like, say, the number of publications that mention a certain volatile, even if you haven't got the quantitative data, that could be quite useful, because you then get a sense that that is probably something that you might want to quantify, if that makes sense.
Yep.
Whereas if you don't have that all in one place, it can be difficult to sort of make those decisions if you've just got the data that's been quantified. And I noticed, in the Atlas, there's like the disease associated data, but obviously the actual data there, I'm assuming, is like in terms of the healthy, like baseline, in terms of quantification of the data. So I don't know whether there's plans to sort of like add in, I think there's possibly plans to add in like the ranges in diseased versus healthy, for example.
Yeah, there is for sure. So obviously in retrospective, what's been reported before, that, if it hasn't met the quality standards, probably shouldn't go in. For prospective studies, I think our goal was to start with nominally healthy subjects first, because that was exactly the thing that we felt would be most beneficial to us. So when we were running, a disease study for a partner, they may be constrained in terms of the number of samples that they could collect, either through, budget or logistics, or whatever the case may be. But we realized that actually, if a VOC or a set of VOCs came out from that study and it looked like it was interesting, actually, if we could compare it to, a much larger population of nominally healthy control subjects, if you have something that's genuinely interesting, or if it's more likely just statistical fluke from a small sample. So they will be added in time. And that's one of the benefits, I think, that we can have when you build on top of the healthy baseline data. And just the last point before I turn over to Iain, picking up on a point Iain made, we do think it's important to include prospective data for breath and in vitro and animal studies as well, because again, if you're trying to build the strongest possible evidence base for a hypothesis for a compound or set of compounds, seeing similar directionality across different sample types and across different studies is certainly going to be helpful in giving you the confidence to then say, okay, if we run a prospective study with a targeted method looking at these particular compounds, you're more likely to be successful there. So I do think having these extensive data sets will help there. Iain, any kind of additional thoughts that you want to add there?
No. I mean, both of the things you mentioned sound very useful. So it is difficult to just choose one. I suppose if you're reporting biomarkers that have been highlighted as being significant, then alongside that, different metrics need to be reported, like the statistical approach, whether there was external validation, percent specificity, percent sensitivity, that sort of thing. Whereas for the quantitative side of things, it's the analytical approach you've taken, isn't it? And that, as I said earlier, that's I guess what excites me most. Being able to actually scrutinize the different platforms, scrutinize the different studies in terms of actual breath concentrations. I think that's not only important in terms of comparing different studies, but also overall assessing the clinical utility of a VOC.
Yeah. That's interesting. As if you think as well about the data that is downloaded, it is inherently tied to the analytical platform as well. So Julia mentioned earlier, part of the goal is to ensure that when data is downloaded, you don't just have summary metrics for concentration ranges. You can actually see if a sample was below limit of quantitation, for instance. So the user can look at the whole data set and say, well, actually, these are bits that I have more confidence in versus different concentrations. So we've got time for last, and it's a question I ask every year, which is you look 5 years into the future, and obviously standardization has been completely sorted once and for all. But I'd love you to talk in the last couple of minutes just about the applications where you think breath has the best chance, and whether or not that is going to be through kind of classic biomarker discovery. You run the studies, you find on breath compounds, endogenous origin or whatever, or is it more using probes, so things that have worked in the past. We have hydrogen methane breath tests where you have a carbohydrate substrate, or some of the stable isotope tests. So I'll kind of get you to put on your prediction hat and say what tests will be used by patients day in, day out in 5 years' time that don't exist today. And I will let whoever wants to go first on that one.
I'd just like to say that I'm an optimist, and I do believe that there'll be a great sensor technology that will come along and will be able to target the VOCs that we're finding from these biomarker studies. So maybe it won't be a high technology readiness level in 5 years' time, but maybe we'll have got a lot further down the line and closer to clinical deployment.
Do you think, building on some of your research interests, some of the exposure or exposomic type applications will progress?
I'm absolutely, I very much hope so. And it seems like a few people are coming together at the same time with that way of thinking. It's perhaps flipping it on its head a little bit, but we know that, for example, what you're exposed to might be a confounder in a study, but yeah, let's flip it on its head and actually use breath to biomonitor.
Makes perfect sense. Brooke, do you want to have a crack?
Sure. I guess my hope or vision is maybe a little bit more generic, but we've talked a lot about specifics of, identifying this infection or that exposure. And I think, some of our ideas are more in a, kind of threat agnostic space. So if we know what normal is, we don't necessarily have to know what specific thing that you're infected with or have been exposed to, but we know that something isn't right. And so I hope that we have a better sort of just idea and understanding of breath so that we can start to make steps in that direction.
Yeah, no, that's great. And knowing what normal is, as you say, is the key to being able to detect when something goes awry. James.
Just like, I remember sitting with you in Leeds 10 years ago and we were discussing this exact question.
Yeah.
So my kind of feeling is that if we're going to move it into a active clinical setting, it's likely that where it starts off is where you either need to make an immediate decision. So I think infection is going to be a big thing, RTI and associated bits and pieces. I think that anything where if you get it kind of centralized lab and you get it wrong, where the risk is really high, I think that will take longer. I think they'll be more like 10 years. But those conditions where if you get it wrong, they're still alive, a week or two later, I think that they're the diseases where I think there'll be really, big uptake. So point of care technology, we'll have kind of some sensors, which will probably be either kind of metabolic disorders, what you're attacking, gastro-related or infection, and then centralized where, the GP is collecting a sample or shipping back for non-life-threatening conditions.
Yeah, that's interesting. Since I don't know if you're in the session yesterday, but the OMED device that we have now is targeted towards the GI conditions. I think exactly for a lot of the points that you just raised there, James. So yes, since our last chat, we can send you a few of those to have a play with as well if you want to have a go.
Oh, that'd be great.
Ben, you have the final word.
Yeah, I mean, I think I've listened to some of the talks and I've been quite heartened by the fact that there seems to be more work penning down like the underlying mechanisms for some of these things and like linking specific volatiles to actual infectious disease, for example. And that, maybe not 5 years' time, but that gives you some hope that you can maybe target some of these like TB, malaria, various infectious diseases such as those, but also, understanding why these volatiles are formed is the most important aspect in terms of like really sort of like targeting. detection methods, etc. So like, especially if you're looking for a multitude of markers to pin down a single disease, for example, it makes making a system quite difficult for that. But there are some potentially more simple, like single volatile targets, potentially, which would make it more viable, I suppose.
Wonderful. And with that, I want to say a massive thank you for joining us today on the panel discussion. It's been a real pleasure, and thanks so much for the contribution today.